All right. Good afternoon for everyone who's watching us today. First of all, I would like to thank Professor Jarquin for being available to participate in our event and for sharing some of his knowledge and experience with us. Feel welcome, Professor. Just a quick resume about Professor Diego Arquin. He holds a bachelor degree in statistics from Chapingo Autonomous University and both master's and PhD in statistics as well from the graduate school in Mexico. He completed postdoctoral training in biostatistics at the University of Alabama and at the University of Nebraska, where he later served as a research assistant and then associated professor. Since 2022, he has been an assistant professor in the agronomy department at the University of Florida, working on statistical learning and multi-omic integration for the prediction of complex traits and the genotype by environmental interactions. Professor, we like to give you the space and you should start the presentation now. Thank you very much for your time and you're available.
Okay, thank you, Marco, for the nice introduction. Also, thank you, Rafael, and the organizing committee for this invite. It's a pleasure for me to be here. So I'm going to start sharing my presentation now. Can you see my screen?
Yes, Professor. Perfect.
Okay. Hi, everybody. Welcome to my presentation. It's an honor for me to be here. Today, I'm going to talk about this multi-omic integration for leveraging G/E in applied prediction breeding. So this is the outline of the presentation. First, I'm going to talk a little bit about deep learning optimization. You know, now with the boom of AI, people think that we can solve many of the G/E problems just adopting those methods that were developed for other implementations. And then to fit in the context of this event, I'm going to present Chido. Chido is the software that we developed to integrate in an easy way all this complex data in order to perform predictions and estimations of the interactions. And finally, I'm going to talk about scaling G/E using GPUs. And this was a motivation because recently there was a paper for one of these groups that presented a new method that they criticized a little bit. The model that we developed is good, but it might be very time consuming, et cetera, et cetera. And then one visiting a student from Brazil took the initiative to develop this new method for solving these heavy, complicated questions in an easy way. So first, I'm going to talk about deep learning optimization. As you know, several students have been published for genomic selection using deep learning methods. In general, some of them have shown moderate to no significant improvements with respect to the conventional model, let's say G-block model. But in any case, the AI is increasing our expectations for significant improvements in predictability. Again, we are using methods that were developed for other type of problems like healthcare, engineering, chatbots, and we think that it's just about borrowing the models and implementing in plant breeding problems. So we know that at least in deep learning, the architecture depends on multiple factors that we call hyperparameters that we have to establish this before we start with the analysis. And we have, for example, number of hidden layers, activation functions, neurons per hidden layer, learning rate, epoch, batch, dropout, etc. Regularization parameters. So to our knowledge, and I might be wrong, I haven't seen that many studies presenting a consistent comparison of systematic comparison of all of these different levels for the different hyperparameters. On the other hand, we also have the genomic selection depends on many factors like LD, the quality of the phenotype, trait architecture, etc. So the motivation of this study was to study to evaluate the different hyperparameters in deep learning because we were interested in finding clear patterns. What will be the most important hyperparameters that we need to consider for moving ahead and improving predictability, and what are the not important or less important hyperparameters in the deep learning context. So this is a study from my student, Ravindra Garapati. And here we consider, as part of the deep learning architecture, different levels for dropout, learning rate, regularization parameter for lambda, neuron layers, number of neurons in the second layer, number of neurons in the first layer, activation function, this vary from layer to layer, in layer 2, layer 1, the batch size, number of iterations or epochs, and inner replicates. So usually we need to replicate many times within the same initial training testing set. So it seems like we have very few levels for all of these hyperparameters, where when we consider all of the potential combinations, we end up with something like 5 million combinations. So at the beginning, we were hoping to consider at least 10 different levels per hyperparameter, but that would have been something that we won't be able to handle. So we consider two different cross-validation designs. For this, we use a data set from a soybean population. This soybean population has been studied already many times. It has 1,379 genotypes tested in 6 environments, all genotypes in all environments. We have 3 traits. The most important, of course, is yield. And the 2 cross-validation scenarios that we consider was prediction of new genotypes and incomplete field trials. So here we consider 5-fold cross-validation, and considering all these elements for the cross-validation, 3 traits, 3 replicates, number of folds, we end up with something like 90 different cases. So in this case, the G-block model, based on main effects, was used as cornerstone to contrast the results. And also we add the G-by reaction norm model just to have an idea more, to conduct a more fair comparison with the deep learning approach. So these are the results. So you can imagine 5 million different results might be a little difficult to analyze in order to make sense or to find clear patterns. So for this, we consider a very simple linear regression where the response was correlation that depended on the common mean plus the activation function combination, because we have 2 different hidden layers, number of epochs, batch size, number of neurons in node 1, number of nodes in hidden layer 1, and number of nodes in hidden layer 2. Regularization parameter for lambda, dropout, and learning rate. And what we found is the activation function combination was the most crucial element, and then followed by learning rate and epochs. It means the batch size, dropout, regularization was not as crucial as these first elements. Based on these results, we presented the results where each one of these vertical, orange vertical lines represent the different activation function combinations. As you can see here, it's very clear that there are some cases where the activation function is not good, and some cases where the activation function is good. So we have at the beginning those that involve the logistic regression as activation function, and in the right side we have those that consider either the tangent or the ReLU activation function. We have also within each one of the orange vertical lines the learning rate, and it's clear that as the learning rate increases, predictability increases. So in this case, predictability is in the y-axis, and within that we also have vertical gray lines that correspond to the number of epochs. In this case, the more epochs, the more we improve predictability. So those results were for the problem of predicting new genotypes, and these correspond to the problem of predicting complete field trials, where some genotypes were already observed in some of the environments. In this case, it's clear to see some clear patterns, right? As we use one or another activation function, we can improve predictability. As we increase learning rate, we improve predictability. Also, the batch size is important, and the number of epochs. So, to make a more fine analysis of these results, we just keep the last three combination of activation functions, where we have ReLU and tangent activation functions, and we divide this by the number of, and we just keep those cases where we have learning rate of 2 and 3. So we discard the 0.1. And within this, we also consider the batch size, right? The subsample to fit the models. And it's clear that as we increase the batch, as we decrease, in this case, the batch size, we improve predictability. And within that, we also have the case where we consider the number of neurons in the second hidden layer. And it's important, it's very clear to see that as we increase the number of neurons in the second hidden layer, predictability improves. So in this case, as you might, let me see the point. Okay, you can see the keeps increasing. Probably if we add more neurons on the second hidden layer, we can keep improving even more. So this is something that we are considering for a second study, but the idea was that, right? Whatever is not presented here, like regularization parameter, dropout, etc., is condensed in this box plot. So here, low batch size return the best results, right? And the most crucial result in this case is that whatever happened in the second hidden layer is more important than in the first hidden layer. So those were the results for CV1, and for CV2, we have something similar, so mixed results. And then we contrast with the standard, gold standard, the G-block model. For yield, it was the only case where we observed significant difference, right? Consider the model with main effects, gave us an average correlation of 0.4. The base case, in this case, the box plot, returned for 0.47 for CV1, 0.41 for CV2 versus 0.48. So it's clear that deep learning method works better than main effects, but this might be a little unfair comparison because we know by the fact that the deep learning is modeling the interactions. So for that, we consider the GBI model, the reaction norm model, right? And then we found that actually deep learning was not that superior than this G-block fashion for modeling the interactions. So we have 0.54, 0.57. And then we went beyond the comparison. We said, let's say that we can pick the best combination across all of the multiple unique cases, what we can call the best case, the maximum of the base case. So in this case, we got 0.53 with deep learning versus 0.54, 0.53, and 0.57. It's getting there, but it's not better than the reaction norm model. So in this case, we will explore what will happen if we keep increasing the number of neurons on the second hidden layer. So hopefully we can somehow beat this cornerstone model. So the conclusions is the deep learning is a powerful alternative dealing with larger assets. However, we have to conduct this optimization process, and it should be exhaustively conducted. The good thing of this study is that now we can discard those hyperparameters that might not be important for this trait and for this crop. And then we can focus on optimizing only those that are important, like learning rate. For example, we found that it saturates when it's close to 2. And also we can discard some of the activation functions that might not be important, and focus on only those that are okay. So again, further studies will be conducted to evaluate what happens when we increase the number of neurons on the second hidden layer, and also then what happens when we increase the number of hidden layers as well. At the end, the idea is to develop a sparse algorithm optimization to find the best strategy by avoiding the computational burden. So it's impossible that we can conduct all of the comparisons with a very large number of levels for the hyperparameters. Okay, now I'm gonna switch gears, and I'm gonna talk about Chido. In this case, it's a software that we developed. First of all, Chido means cool in Spanish. Some Brazilian told me there's something like Isocara. I don't know if that's true or not. So it's a non-code application that aims to remove technical and financial barriers associated with modeling genotype-by-environment interactions in a prediction context. You know, we listen in the morning about very sophisticated models and implementations and technology and using satellite information, etc., etc. But many times in countries developing, we have either new companies or new breeding programs want to test or want to use the new methodologies, but they don't have enough resources to hire a specialist in quantitative genetics. They might have the breeder. They need somebody with skills for programming, statisticians, etc., etc. So the idea was to provide to the users something that they can just plug in their own data and evaluate how good or how bad that data might be for a future context in predictions. So Chido stands for Characterization and Integration of Driving Omics. And this idea started initially with a master student, Francisco Gonzalez, who currently is a PhD student at University of Oregon. And he works as model apps lead at Google North America. So the second student is Julian Garcia Vadillo. He was a PhD student, a visiting PhD student with me last 2 years. And then he moved to work to Google as intern. And currently we have one of your conterráneos, Victor Segage, is a PhD student. He's a visiting scholar from University of Viçosa, and he is the one in charge of the new implementations of Chido. Unfortunately, he's going back to Brazil to take a role as a breeder in a big company. And this is the idea of how we integrate these multiple omics. So let's say that we have an omic based on genetic data or genomic data, and then we can multiply that omic by its transpose. So we can create a genomic kernel, right? If we have another omic that could be taken at the plot level, right? We have something similar. We can create a kernel by multiplying the omic by its transpose. So if we have replicates, obviously we need to use some incidence metrics to redistribute the kernel across all of the phenotypic records. And we can use results from the reaction norm model that we developed some time ago to create the interaction between every single level from the first omic and every single level from the second omics in a way that we end up with a covariance structure. So obviously we use assumptions of the multivariate normal distribution where we add a specific variance component for each one of the different terms in the model. And this can help us to evaluate how good or how bad or how important each one of these different terms in terms of predictability or to explain phenotypic variation. We can play around with the different components to compute things like heritability, for example. So regarding the model evaluation, so as I said before, we look at the variance components and different prediction scenarios. So far, Chido has included these 4 prediction scenarios: sparse testing designs, predicting new lines, predicting new environments, and predicting new lines in new environments. So in summary, Chido is a shiny app that implements the concepts that I explained in this presentation. So the first thing is check data integrity and connects omics data using a specific ID keys, build covariance structure, and design the matrices to model main interaction effects, generate the cross-validation schemes, and collect the predictions and perform the performance for the metrics, right? Currently, we are conducting the forecasts of trait performance using hybrid prediction, host pathogen interactions, and stability studies. I'm going to show a few of these things in a moment. So the next release will allow the GY analysis, sparse testing designs, deep learning, etc. How can I have access to Chido? So you can access on demand using this link. And this is how the Shiny app looks like. I'm going to explain later a little bit more specific the different inputs in this case. Or you can download Chido in your local computer. You can just go to our GitHub account and download Chido on your computer. And this is the interface. So we have an interface for adding the data, another to compose the linear predictor, another to choose the cross-validation scheme that you want to consider, and another for review the results. So in this first interface, we can pick what type of studies we want to conduct, genotype levels of conventional cross-validations, combinability to conduct the prediction of hybrids, and host pathogen interactions. So after you pick the type of study that you want to develop, you just have to provide some specific information, like which is the column that has the ID of the genotypes, which is the column that has the ID of the environments, or whether you have a specific ID column. For example, with phenomics, you have a specific observation for a genotype in an environment. And also you specify which is the trait that you want to analyze, right? What column has that information. On the next interface, you start selecting the data. You can add as many omics as you want, and you have to link those with a specific column, right? That you are going to use as reference to connect with phenotypes. And then you can rename the label. For example, you can use weather data. You can just call W, or you can use C or EC or environmental covariance, etc. Right? And then for those cases where you have a specific or a compound ID for genotype in the environment, you also select that information. And this is my favorite part, how you compose the linear predictors. And it's a drag and drop, right? You can just drag the information of the available omics and drop in this case and click add to add the components. You want to add an interaction, for example, E and W, you can use drag and drop these two components. They will collapse, and then you add to the model. If you don't want to play like this, you can just type directly the linear predictor in this box for the formula. So something like this. And then we conduct the analysis. So we have to specify a few things like the hyperparameters for tuning the model, number of iterations for burn-in rate. And also we can specify the number of faults that we want to simulate for the different cross-validation. You want to apply quality control to the data. And also we recommend to standardize and center the covariates, because you can have a more fair comparison of the importance of the component when you look at the variance component. So you pick the different cross-validation schemes you want to analyze, and then for each cross-validation scheme, there are 6 steps associated, and then you will have that information of the progression in one of the boxes. For example, if I want to use Chido for hybrid prediction, right? We know that many of these breeding programs focus on development of hybrids. They have to evaluate a very large number of single cross combinations, and the selection is being conducted initially within the different heterotic groups, considering all these different combinations of different heterotic groups is costly for sure. It might take a lot of time. And on our favor, we have that we can use molecular markers. So we can cluster inbred lines into heterotic groups. We can predict the performance using the general and specific combinability terms. So this is the data format, the input. that we need to run this in our implementation. So you need a column that has the ID of the environment, a column with the name of the genotype, in this case the hybrid, a column with the response, and a column where you have the ID of parent 1 and parent 2. So this hybrid is composed by this combination, right? If we have missing values, so we want to predict a cross that is not already observed, we can add any 8 in that case. Okay, so we have to select the combinability option, right? And we add all this information that I mentioned in these boxes, right? Genotype ID is column number 2. Parent group ID, 1 is column 4, 4 and 5, and so on. So regarding the input of the data, it could be a pedigree metrics, right? That could be for all of the genotypes that we are analyzing, involving in the process of the hybrids. It could be marker information from the inbreds, etc., etc. So it could be, it's very flexible. We can add any information either as a covenant structure or as a set of covariates. And then we pick the different omics. I said genomic markers, phenomics, environmental markers, pedigree data, high throughput data, and others. So it's very flexible. So, and then we can add also what will be the linkage for the different components in the model. Similar than before, we will have the components, for example, environments, line effect of parent 1, line effect of parent 2, marker information on parent 1, marker information on parent 2, and we can elaborate very complex models in this case. And this is the same as before. We train and validate using the different values for the hyperparameters. We have the models, the cross validations, and then we obtain the results, right? We can visualize the results within implementation, or we can download the results to create our own plots. So in this case, we will get the results for the different cross validation schemes. First, at the within environments, and also as a weighted average across environments. So that will be for predictability as a correlation, and also considering the mean square error between the predicted and observed value, and also the variance component, right? In this case, we can observe the suspected environment captures more of the variability, and then we have the different components in the model. This model, this implementation also allows the interaction between parent 1 and environment, environmental covariates, parent 2, and the same between the parents, and it could be as complex as you want. Another implementations that we have is GY estimation stability parameters. So this is a extension that was conducted in collaboration with people at the University of Viçosa. So with Professor Moises as the main lead author of this study. So as we learned during the day about GY interactions, how important it is to analyze, to estimate whether the interaction is good or not, because we have to perform recommendations, right? The stability, high performance, analysis of multi-environmental trials, etc., right? So we learned that some cases it might be very complicated to conduct a recommendation. Sometimes the models might be very difficult to fit. In this case, we just focus on the EuGeM model as the base model to develop the other indexes. And the model is the model that is based on the Bayesian approach. So this is a paper that we published some time ago, and then we extended, in this case, to add these new indexes. So Chido already integrated the following approaches: the Bayesian EuGeM model, Bayesian version of the frequentist approaches of WAS and WASB. So these indexes are important because it's not just about developing a genotype that is stable. So it should be stable with high performance. So we can weight the importance, right, of these two factors to conduct the recommendation. And the new Bayesian stability index is going to be based on Mahalanobis distance. This is the proposed study by Dr. Moises Nascimento. So this is the format to enter the data in the model, right? We have a column with the ID of the site, another with the name of the ID of the genotype, replicates whether we have or not, but we still have to have that column, and the response. And then we conduct first the Bayesian analysis based on the Bayesian naming model, where we have to provide information on the number of iterations, total number of iterations for burning rate, tin, right, to avoid this high correlations between iterations, etc. And then once we finish the analysis, that we can pick what are the analysis that we want to conduct based on the output from the Bayesian aiming model, right? We have stability, WAS index, WAS site index, and we pick what the importance, right, of the different factors. We want something that is going to be stable versus something that's going to have high performance. And these are some of the results, right? First, we have in the top right the results of the Bayesian IME model. In this case, we didn't include the biplots. And also we have the traces, right? Just to make sure that we are reaching convergence. We consider the WAS index to check the stability of the genotypes. We get an output like this, right? Low values means more stable genotypes. That's one output. But we might consider also high performance. In this case, higher values means more stable and high yielding genotypes. So as you know, in the biplot, the closest to the 0.00, the most stable, but might be the case there's a genotype that has a low performance. Further we go in one or another direction, means that it's less stable, but we don't know if the performance is high. So we have to weight these two sets of results. Also, we have the step this, where we can analyze the outputs based on quadrants. In this case, those genotypes in the fourth quadrant will be the more consistent based on stability and performance. If you need, or we are interested in more details about this technique, I really suggest you to contact Dr. Moises Nascimento. He's the one who knows the specifics of this set of analysis. So what's next for Chido? As we saw, Chido is a powerful tool to conduct estimation and prediction analysis. It's a message that we wanted to give a long time ago is that this implementation is not exclusive of breeding applications. We can use in other applications like personalized medicine, where we have the same elements, markers, habits, social context, etc., etc. All of these things will somehow interact. We already had interaction with pathogens. We will be moving forward with the optimization of the allocation of the resources with some sort of sparse testing design. And we are going to integrate these AI methods, right, in a way that we can consider a more informative set of results. So the main goal is to develop an open source suite of applications that will be easy to adapt, easy to use. And also we are open to collaborations or partnerships. So obviously we don't have the answers for every single question, and we don't have all of the techniques listed there. So we welcome that. So finally, I'm gonna talk quickly about scaling G by E using ZPUs. As I said before, recently there was a manuscript that somehow criticized that, okay, reaction norm model is good, but it might be very complicated to implement in real life experiments where we have large data sets. So Víctor Segage came up with the idea of trying to develop something that can help us to tackle this problem of having large dimension data. So as we know, AI is a set of techniques that are creating a revolution in the past years, has changed completely many of the different applications in the broad group of fields. And it's because the methods have been improved or developed to come up with these new inconsistencies. So we can use now graphic processing units to execute highly parallelized operations. So here is a brief comparison of using CPUs versus using GPUs. For example, if we want to consider an operation matrix of 100 by 100, using one CPU can take 0.36 seconds. Using the GPU might take 0.3. So using the small scale problems is not where we are going to see a big difference. The big difference is going to be when we use a large scale problem. For example, if we have a matrix of 5,000 by 5,000, it could take 9 minutes to conduct the analysis using the CPU. If we use the GPU, it could take 1.30 seconds. So this is the advantage of using the GPUs. Also, we know the AI has been implemented for predicting in plant breeding. Despite of being very promising tool, we found that actually it's not showing big difference with the conventional implementations that we have, like gblob, rblob, RKH, Bayesian alphabet. And also these models the implementation of these models has not been optimized. It's one of the problems. It takes a lot of time to run analysis in this using this type of parametric models. So I'm going to explain a little bit about the challenges working with highly dimensional data using complex interactions. So as you know, if we consider a parametric model based on a linear combination between marker and marker effects, usually P is greater than N. And in those cases, we cannot get the inverse of these metrics to get the solutions, right? We have to add a penalty function to be able to break this singularity. We can do that with the penalized regressions and with the Bayesian regressions. Okay, we can get a set of results. However, we're going to be dealing with highly dimensional matrices of order P by P. So if we switch around and we, instead of working with the linear combination, and we start working with a covariance structure, right, that represents the genomic effect of that linear combination, we can, using a kinship matrix, we can break the singularity very easy, and then we can deal with matrices of order n by n. And this is fine, but what will be at the future the problem when n starts increasing? Some time ago, we come up with this idea of also introduce interactions as the interaction between different levels for the different factors. And for example, if we have two sources of variation, and we consider the contrast, we have the include in all of the contrast in the models can be very complicated. If we have, for example, 2,000 markers, 100 environmental covariates, the number of contrasts to add in the model is 200,000. We already had those problems before with just main effects, and now just imagine adding all of the single contrasts, right? So, okay, we developed this reaction norm model where we consider a specific set of the scores, and we consider the multiplicative mode, and we were able to use or to show that actually using the product between the cell-by-cell product between covariance structures can help us to deal with that problem. Now, what happens when we have replicates across environments? We can use, for example, the incidence metrics to redistribute that covariance structure across all of the different phenotypes. But the main question is, but what in this case, n is also large. So we were dealing a little bit with how to reduce dimensionality using a very large number of predictors. But now what happens when we have a very large number of phenotypic observations. And this is an example. If we consider we have 2,000 genotypes observed in 100 environments, the total number of data points, the result of this is 200,000. If we have a covariance structure, it means that we are going to end up with a matrix of 200,000 by 200,000. We need 8 logical bytes to store each one of these cells of the matrix with a double precision. That is what R uses. We need 320 gigabytes of RAM memory to have that information at least available for analysis. We can even consider a symmetric matrix where we can use half of the matrix, right, including the diagonal. So the memory required to load matrix increase in a quadratic scale, right, 0 to n squared. And usually for genomic prediction, we are expecting to have more than one matrix involved in the fitting. So we can have interactions, so we can have more omics as covariance structure. So the objective of this study was to employ new tools that have been used for training lab datasets for AI models in traditional parametric approaches for genomic prediction. In this case, execute expensive linear algebra operations in GPU instead of using CPUs. And this is a very nice representation that Victor put in the slides. So, and the analogy that he told me was very interesting. He said, just imagine that you want to discover the cure for a bad disease. You will pretty much select the top 10 scientists around the world, and then you will ask them to work together. They say that will be the case of the CPU. He said, on the other hand, suppose that you want to build a football stadium. The more people you have working in the stadium, the easiest. So consider that will be your GPU. So in this case, the data set that was analyzed is comprised of 2,500 genotypes that were evaluated in 340 environments between 2015 to 2018. We have very few markers, less than 3,000. Total number of combinations of genotypes in environments, 850,000. And we use a HPC node from NVIDIA with 2 CPU cores, 120 gigabytes of memory, one NVIDIA Core A100, 80 gigabytes of memory, and libraries from Rapids, QDF, and CuPy. So Victor can provide very specific information about the different libraries in this case. And this is the core of the implementation. So suppose that we have a matrix of molecular markers for G genotypes and P markers. And consider Z sub G, the incident matrix, to redistribute those markers across all of the environments for the genotypes. And then, if we want to construct a covariance structure, we will proceed as multiplying Q by Q prime. But let's say in that case, we can compute Q prime by Q, because we know that we won't have more than 3,000, a matrix of 3,000 by 3,000, instead of using 850,000 by 850,000. So following these results of the PCR, we have the Q prime and Q prime share the same non-zero eigenvalues, and makes sense. We are just switching around the information. So in that case, we can recover the eigenvectors of Q prime, that is the covariance structure that we are interested, by using V equal to X, the matrix of molecular markers, times U, and the diagonal of the inverse of 1 of the square root of D. This process is going to allow us to obtain the eigenvectors and eigenvalues from the kernel that we are interested, ZG prime, without having to explicitly to build this matrix. So we solve that problem in that case. Now, working with interactions, right? Because we might be interested in G by interactions, we can apply a similar trick, right? Starting with the G by G matrix, that is decomposed based on the eigenvalues. We can reconstruct the matrix using only a few principal components, let's say 80% of the principal components. So the principal number of principal components that explain 80% of the variability, cumulative variability, and then we can build back the Q metrics, right? In that case, it's going to be quite easier to compute the metrics that include the interactions. And these are the models that were used, a model with main effects and a model with interactions. So these are the posterior densities. And for the solver, we use the joint posterior using a Gibbs sampler, right? We know the Gibbs sampler is highly sequential process where the next iteration depends on the previous one, but this process can be accelerated using the GPUs. So in this study, we consider 20,000 GPUs, 20,000 iterations, 4,000 uses for burning, and T equal to 4. So, and these are the benchmark. We consider different sizes just to see how the requirements of computer requirements increases. So with a data set of 100,000 records, it took around 5 minutes to fit the model. When we consider 2,000 records, it took 40 minutes. So it increased by 2.3, right? The requirements. Using 4,025, it took 30 minutes. So it's now the requirement multiplied by 1.7. And when we reached the 850,000, we were surprised. So the requirements remain quite the same, so 1.7. So it took less than 1 hour to fit the model with 850,000 records, considering the G/E model, and a little bit more than 40 minutes with the main effects model. So it's not even that bad, including more omics in this case. So we can look at the variance components at the traces. So we make sure everything is reaching convergence. And then we tested based on predictability, right? Forward prediction. In this case, we have 2 different training sets. In blue, 2015 and 16 environments, and in yellow, all of the environments between 2015 and 17 to predict 2018. So left panel, we have the model with main effects. Right panel model with also includes interactions. So the models, the environments are ordered based on predictability. So this was the environment that was more poorly predicted, and this environment that was more highly predicted. So it's clear that when we have a larger training set, it's easy to predict with high confidence. As expected, predicting new environments when G/E model is not that a good idea, but still the results look okay. Predicting 500 new genotypes in environments that were already observed is a different story. So here we again order the environments based on the predictability, and it's clear that the model that includes the interactions always, or most of the time, beats the model without the interactions. So it's important to note that actually now we can work with very large data sets for training the models instead of sacrificing some data by randomly select the phenotypic records that will be part of the training set. So in conclusion, we had that the use of GPUs for fitting genomic prediction models considering G/E interaction can help breeders to improve larger data sets. And this is something that we already observed in other areas like animal breeding, where they have very large number of individuals, but they have to sacrifice the training set by selecting randomly some of the individuals for training. Scalability will always be a challenge despite the computational power is evolving. So now with the new methods that have been developed for AI, we can take advantage of those, right? The libraries, how to handle large data set to use in the conventional prediction methods that we have. The reaction norm model is a useful alternative for modeling the G/E and integrate multi-omic data. Despite the higher computational cost, GPU can help to overcome these limitations, right? It's important to note that GPU programming is evolving very fast. So now we have new GPUs that are easily 4 or 5 times more powerful than the GPU that was implemented by Victor to run this analysis. So I think moving forward in the future, we need to consider or still consider these parametric models to conduct the analysis. Of course, we also have to keep doing research using the AI methods. The cloud service can turn this technology more affordable in the future. With this, I want to thank people in the lab, people who supported and helped to develop many of these implementations. And of course, as always, Go Gators. And if you have any questions, I'm more than happy to answer.
Hello, Professor. Thank you very much for the presentation. We have some questions, but first of all, Chido here in Brazil is like top or bacana, you know?
Really? That's good. Good to
I
know.
was thinking about some correspondence word as we can use here daily, like you use Chido informally, you know?
Oh.
So we have a few questions, okay? First of all, question from Tassinari, Rafael Tassinari. Diego, my question is, how can GPU-based algebraic operations be integrated with RML workflows, including matrix inversion?
Oh, this is a great question, and I think the more qualified person to answer the question might be Victor. So he is the one who has been working directly with these new libraries from NVIDIA, CuPy, and QDF. But to be honest, I don't know how that works in the context of connected with the GPUs.
Okay, perfect. After we have two questions from Khalid. First of all, how can multi-omic data, genomics, transcriptomics, proteomics, metabolomics be integrated to better understand and predict genotype by environmental interactions in breed programs? And now I will add like environmics as well, because is what we work here in LAMP. So how can multi-omic data can be integrated to better understand and predict genotype by environmental interactions in plant breeding, in your opinion?
Yeah, I think we have to start with differentiating. So we have in statistics two different problems. One is estimation, where we are really interested in learning what is the underlying conditions that are affecting a specific trait, right? It could be like basic research, like going and see pathway analysis, GWAS studies. That's one part of the question. The other part is prediction. And having a very large data set and multiple omics, seems to me it's going to be more difficult try to understand what is going on with the different pieces, right? Moving forward, predicting. For example, environmics, yeah, interesting to have all that information, interesting to be able to improve predictability, reduce mean square error. But I don't know how much inference we can make based on the environmental data that we are collecting versus going to estimate what is the effect of a specific weather covariate, where the most important period of time where temperature is affecting yield performance of flowering, etc., etc. It sounds difficult. Now, Chido, for example, offers that flexibility that you can add as many sources of variability as you can collect. And probably the predictability will keep improving. I don't know how much inference we can do or perform going back to see what is the specific covariate is causing those variabilities. I think the most that we can do for now is just go and look at the variance components to see which of the different components in the models are the more important. And go moving forward, just isolate that signal and now go and perform a more specific analysis with the specific layers.
Okay. Other question from Khalid is, what are the advantages and challenges in incorporating multi-omic data into GBI analysis for predictive breeding models?
I think from a point of view from the user, one of the most difficult challenge will be collect the data and make sure the understand that more data doesn't mean more signal. It could be more noise too, right?
Yes.
And I think the main question using, for example, environmics, as Dr. Piepho said this morning, yeah, I can have a model is really good predicting, and when I add the weather information, but I don't know what will be the weather conditions in 3 months, 6 months, next year. So I think there are things that we can control, and the main idea will be like dissect GYE and have those components that are easy to predict, for example, day length, and see if that information can add a lot of value to the results versus trying to integrate things like temperature, right? Because we can have a specific event where temperature can increase, I don't know, let's say 10 degrees Celsius in a very short period of time, some stress that we cannot predict those stresses.
Okay. Um, One question from Bruna. How can multiomic GBI models can be structured to assign different weights to each omic layer, um, like genomic, transcriptomic, metabolomic, enviromic?
I mean, one natural way to to weight the importance is by looking at the variance components. Right? We will
Mhm.
see how important is one component versus the other. Now, something that we don't want to sell the wrong message is that yeah, works for predictability. However, a lot of this information might be repeated. You have transcriptomics, you have genomic data, RNA-seq, etc., etc. So at this point, we are unable to create right, orthogonal sources of variability. So whatever is not explained by one factor is going to be captured by the other. So it's kind of difficult. And I think at this point, the main idea is to still not explaining signal from the residual. Later on, probably we can focus on create this orthonormal, orthogonal contrast.
Okay. A question from Bruno. What modeling strategies are suitable for estimation G by E effects under highly unbalanced and sparse genotype by environmental matrix?
But that's a tough question. I think
Mhm.
this is I wish I could say things like Amy model, but we know this should be a perfect or at least balance. I think the most suitable techniques that we have are Finland Wilkinson regression or the method that were today explained by Dr. Piepho.
Okay. Last one. How does the choice of genomic kernel, like linear, Gaussian, and arcosine, influence the stability and interpretability of GBI interactions kernels derived via PCA?
It's interesting question. We know that using these AI methods or semi-AI methods or methods that internally model interactions might improve slightly predictability versus main effect models or parametric models. I think one of the main challenges there is to understand that we know that parents pass alleles to progeny. So very difficult to handle, right? To pass the whole genotype. So in those cases, also sounds very difficult to try to influence how those interactions will be passed to the progeny. So that kind of are the challenges that I envision. That so far we have good machinery to perform the predictions. Moving forward, I don't know how much we can influence in that sense.
Fine. Okay. Professor Rafael sent me a message here. He's very interested in GPU-based algebraic operations, particularly for GBI models. and he said that he's working with DF Remo rather than AA Remo, and have achieved good results using optimization routines. And that's complements the question he asked earlier. Okay. Professor, I would like to thank you very much for your time and for the available you have been for respond these questions and participation this year in GBI Day. Thank you very much. And I would like to thank you all of our viewers since this morning. And I would like to extend the thanks to all the speakers that we had today. And of course, for the team who has organized such a beautiful event and huge event on this holiday there in USA. So extra thank you for that, for taking the time on your holiday to speak with us. And that's it. Thank you very much, and appreciations
No,
here from Brazil.
obrigado. Thank you.